Okay, Assalamu alaikum wa rahmatullahi wa barakatuh. I think we can start maybe in one more minute. نعطي كده بس دقيقة كمان for everyone to sign in, and then inshallah we can start. Okay, Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you so much for attending this webinar. Shukran for King Fahad Medical Research Center and my unit, Regenerative Medicine, for giving me the opportunity to give this webinar. Of course, it's always a bit difficult when it's a very broad audience. I'm sure there's a lot of students and there's a lot of diversity in the specialties. So I really try to make this as broad as possible so that everyone can have a little bit of some benefit from the webinar in one way or the other. Basically, today I'll be talking about how we used patient-derived xenografts and transcriptomic signatures to predict treatment response. And the model I was using was metastatic prostate cancer, but obviously this can apply to any type of tumor. I'm just going to try to make it specific to what we've been doing, but it's a very general approach that we can obviously use, and I'll just go through the methodological things that we've done. To give you an idea about this research context and the collaborators I've been working with, so basically I woke up one day and I decided I want to learn dry lab. I wanted to learn more about bioinformatics, and I wanted to shift from wet lab towards more of data analysis and dry lab. It's never too late, so I decided I'll take 1 year sabbatical, 1 year research communication year. And I decided to do this sabbatical in McGill University in Canada. It was too cold, but it was worth the year. And I've done my year basically to learn a little bit about bioinformatics in the form of transcriptomics. In Dr. David Labbé's lab. Dr. David Labbé is an associate professor in the Department of Surgery, Faculty of Medicine in McGill University. He's also part of the experimental medicine. And his lab is in the RI, Research Institute of McGill University. He's under the Cancer Research Program. And Dr. Labbé is interested in prostate cancer, basically, mainly understanding MYC-driven prostate tumors and the impact of MYC on tumor development in prostate. He published his Nature paper recently, but also a very interesting paper that I would recommend people who are interested in diet and prostate to read a little bit more on. He's done fascinating work with high-fat diet, short-term high-fat diet, and the impact this can have on prostate cancer in the context of mix. I really recommend people to go through this paper. And during this time, I was under supervision of the bioinformatics help through his associate researcher, Dr. Nadia Boufaid, and there was also a PhD student helping with a lot of the wet lab. Another important collaborator here is Dr. Eva Corey. She is a co-director of the genitourinary research lab in University of Washington. And she's basically the supplier of our PDX model, which I will talk about, not only for us, but probably in North America as a whole. So we're going to go through this. To generally give you an idea about how this webinar is going to go, so everyone knows what are the sections and maybe focus on the parts you're more interested in. I'm going to talk briefly about metastatic prostate cancer, like why. And the question is, why did we use metastatic prostate cancer, or why we're interested in this? We're going to go through the PDX model, talk a little bit more in details. What are PDX models? Why are they very clinical relevant? And what criteria we can use to assess responders versus non-responders, because this is a very important question. I'm going to talk about the transcriptomic analysis that I've done briefly. Obviously, these things are very technical, but just a general idea about the things we can do. And finally, I'm going to complete with the work currently being done by the PhD student, Jieshen, which is the CRISPR-Cas library, in order to complement this transcriptomic analysis that we're doing. And you see, it's a cycle, because we always want to remember that we start from a patient and we want to end up in the patient. It's translational medicine that we're trying to achieve here. Now, to start, metastatic prostate cancer. Prostate cancer is generally one of the most common cancers in men in the world and in Saudi Arabia. And around 8% of them become metastatic or show up as metastatic disease, meaning that it has spread, obviously, from the prostate. And at this stage, whenever it's a metastatic disease, generally, there's a lot of biological heterogeneity, meaning that each patient has a very different makeup of this tumor, and specifically also for prostate cancer. And this makes predicting treatment response quite challenging. It's very difficult to find one therapy for all. Another important point that a lot of metastatic prostate become, although prostate cancer is sensitive to hormone therapy, a lot of them become castration resistant, meaning that even if there's low testosterone, they still grow. And at this point, it becomes very limited to find targeted therapies. There's not much targeted therapies available in the market, and it still makes this disease quite incurable and lethal. That's why we really need to think of this disease. We really need to find better models. We want to find biomarkers that can really tell and guide our therapy. And if you check in Saudi Arabia, and this is based on the cancer incident report, the latest in 2022, done by the National Cancer Center and the Saudi Health Council, you'll see that prostate is number 6 incidence in the Saudis. And obviously in male, it lies second. And even at age 75 onwards, you can see that prostate cancer even becomes number 1 ahead of colorectal in this age group. A very important point about metastatic prostate cancer. And this is based on NIH, the National Cancer Institute in the NIH, USA NIH, the SEER report. So you can see once it's localized, once the prostate cancer is localized, your survival rate is pretty good. The 5-year survival rate is excellent, 99%. Everyone does well. Whenever it reaches to become metastatic or distant prostate cancer, the survival rate drops tremendously to 37%. So that's a big drop. And if you look in the Saudi population, what was interesting to see in this 2022 report of the Saudi population, that we have approximately 24.4% of our patients having metastatic prostate cancer. Although globally it's 8%, in Saudi Arabia it's 24.4 metastatic. And this was an interesting point. We just had a talk by Dr. Ahoud from our unit. We were discussing this point. Do we have a problem with late diagnosis? Do people come in late? And that's why the majority of them are in the metastatic phase, or it's the nature of the disease. So I think there's much work to be done to really come up why do we have this high percentage of metastatic prostate cancer. As I go, I will give key messages in each section just to help people get an important point from each section. So I want to focus here. A lot of students come, and I know there's a lot of students attending this webinar. A lot of students come, and they are very excited to work in breast cancer, colon cancer, and so forth, without knowing much about the disease. So you come and ask them, what subtype are you working in? What histology? What does it represent? What are its receptors? How is it in the region? Are there any available local therapies for? So they just think of a disease as a very broad category without looking in depth on the disease stage. And does it still have unmet clinical needs? So we really need to think about this. But this disease seems to be quite unmet, not enough therapies, and we need much better biological models. This takes us then to really thinking about therapies. And the statement, as much as we know about a little bit, but the number is very scary. 85% of preclinical agents that enter clinical trials fail completely to demonstrate any safety or efficacy to gain regulatory approval. So we work a lot with drugs in vitro and in vivo animal models, and 85% that reach clinical trial don't show any efficacy. Those are even the ones that reach clinical trials. Like a lot of things don't even reach clinical trial. And this really puts a question mark. How well is the predictive value of our preclinical models? Are our preclinical models that we use all the time, are they showing good predictive values in terms of how efficient these drugs are? This takes us to why we use PDXs. Before I start on showing the PDXs we've been using, we know that we can use a lot of in vitro models, and these tend to have like cell lines and organoids, but you're really lacking the tumor microenvironment. You're lacking a systemic factor. You're not dealing with an in vivo animal or model that can really show everything in relation even to drug pharmacokinetics and all that. We sometimes use cell line xenografts, so taking a cell line and growing in a mouse, but there's a lot of selective pressure, and they completely lack heterogeneity. They become all the same. You're dealing with the same genotype of these cells. But most importantly, they've shown to have poor correlation with clinical outcome. So it's not a very strong correlation with clinical outcome, which takes us to patient-derived xenografts. What is patient-derived xenograft? So basically, for those not familiar, so it's taking a tumor from a patient and engrafting this tumor as a tissue, as a small tissue in a mouse that is immune deficient. And this is known as the engraftment phase. And once this is done, you can take these tumors into growing and expanding in other mice. And from there, you can do your research on these mice. So why is this good, or why is this a good model? I'll try to convince you along the way why, but generally it retains the histology. So you're keeping exactly the histology of this patient's tumor. You're maintaining its genomic signatures completely. It's very tumor heterogeneity, because you're maintaining tumor heterogeneity because you're taking one tumor from each patient. So this really reflects the patient. You still have the stromal microenvironment, although you're lacking the immune system, so that's an important point, but at least you still have a lot of the stromal other microenvironment there. But the most important point, if you want to come with just one conclusion from PDX, is that it has shown to faithfully reflect clinical response to cancer therapeutic. If, in simple words, if this mouse responds to the drug, this patient most likely will respond to this drug. Of course, there's limitations. They don't have the immune system. There's no intact immune system. You start having a lot of the mouse stroma involved, so you need to be careful about that. But we do have a lot of underrepresentations of various genotypes. So, for example, we might not have enough from our Saudi patients, enough PDX models, or from other ethnicities. So there are some limitations on the amount of PDX representations that we have. Just to further convince you, now this was published in Nature Medicine in 2015, and it was basically one of the largest PDX clinical trials. So they used 1,000 PDXs population. So this is from a large sample of tumors populations. And they use the design which is known as 1 by 1 by 1. So they take one patient, and from there they make one PDX, and they treat it with one treatment. And just to show you the amount of things you can do, obviously, with PDXs. So, for example, these are different mutations. So these patients that have BRAF mutations or NRAS mutation, different type of cancer mutations. And you can see if you treat them with a BRAF inhibitor, 60% seem to be responding to the therapy, while 40%, the orange, were not responding to this therapy. If you then add another downstream inhibitor, downstream of the signaling inhibitor, you can see that the majority start responding to the therapy. And they've seen that this can really completely compare to the results that they see with patient clinical, phase 2 clinical trial. So it really shows that PDXs are very faithful in corresponding to clinical trials and clinical patients. To take you through the PDXs we've been using, so Dr. Professor Eva Corey noticed that there wasn't enough prostate PDXs in the world, not commercially, not in enough groups. So she dedicated a lot of her time to generate this series of prostate cancer known as the LUCAP prostate cancer patient-derived xenograft. And what they've done, that they've taken 261 prostate cancer samples from a lot of patients during these years. And to show you, this is a difficult task because these are metastatic foci, meaning that they're obviously in the liver, in the bone, and so forth. It's not easy to get during surgery. Not many people obviously will undergo surgical once they're in metastasis. So this needs to be by autopsy. So patients that die, you have a committed team of an autopsy pathologist who needs to be there within 4 to 8 hours after death to avoid any ischemia. And the team then takes these samples, and then once these samples are there, so every time they get a call, they have to have this team to really go and try to take these samples. So it's a lot of work. And then these samples are implanted subcutaneously in the immune deficient mice, and they were able to establish at that point 27 PDX models. So from 261 prostate cancer samples, they were able to establish 27 PDX models. So this is only a 10% rate success. It's not an easy job. Once you have these PDX models, you can then do all the characterization, the genomic profiling of all these models, so that you have all their genetic background and DNA and RNA expression. So it's challenging, and you see that to initially establish a PDX, so from the time you take it from a patient to grow in the engraftment phase, the success rate usually is very low, 10%, and it usually might require from 4 to 12 months to establish this. But once this is done, you then expand your PDX. So you take it from one mouse and then you start growing in the other mice. And this has a better success rate of 50 to 80%. And usually it would take 6 to 36 weeks for the tumor to grow. And we consider this as a successful growth when it reaches 50 milligrams. And then from there, it can start growing onwards till your endpoint. So you can try various treatments on this. But it's very important, as I said, to realize that they are very biologically heterogeneous. So their growth kinetics can vary. This is just an average scheme. And to really show you the LUCAP series and the importance of how it gives all these biological heterogeneity. So, for example, these are two PDX models. The first one is LUCAP 96. It's just the number of the patient. And the second one is the 145.1. And you can see here that LUCAP 96, through immunohistochemistry, stains for androgen receptor. So it's androgen receptor positive. But for example, it stains negative for this SYP, synaptophysin. So it's a neuroendocrine receptor. While LUCAP 145.1 was, for example, negative for androgen receptor, so it's an androgen receptor negative PDX, while stains heavily for this neuroendocrine marker. And so this is more of an adenocarcinoma type of PDX, while this is a neuroendocrine PDX. And we also found some of them that were double negative prostate cancer. We call them like they are AR negative and neuroendocrine negative. And why is this important? Because neuroendocrine prostate cancers tend to be more aggressive. They don't respond much to therapy. There's still limited therapies for these subsets. They don't respond sometimes as good as the adenocarcinoma. So it's really important to categorize these PDXs. So the key message too here, that PDXs can faithfully reflect patient drug responses. This has been proven already in a lot of papers that PDX can reflect patient drug responses. It makes them a clinically relevant preclinical model whenever we're talking about translational and precision medicine and to know what drug suits which patient. I think they make a very good model in that sense. You can still see the integration of the genomic with the drug response, and this helps us in precision medicine, which is the essence, obviously, of a lot of translational work. So from there, since we have this LUCAP series, and Dr. Eva is our direct collaborator, what they do is once the engraftment expanded in mice, they take RNA sequence from these mice before treatment, so pre-treatment RNA-seq, and then they treat mice with various combinations. And I can tell you, anyone who wants to try to test a drug on prostate cancer calls Dr. Eva and gets some of her LUCAP series. So it's maybe her only project, but now it's contributed a lot to any therapeutics in prostate cancer. And whenever you test any drug, you can then see if this mice is going to be a responder or not responder, and then try to check the RNA-seq to find a response signature. Now, very importantly, it's very easy sometimes to generate data. With bioinformatics, it's even easier to get a lot of data, but that doesn't mean that it's good data or correct data. And I think touching on details and making sure you're going correctly in every step is the most important thing. And this is what differentiates having something meaningful or publishing in a paper like journal or not. So really you need to think about the details. It's not just generating data. So it's very important to see how are you going to say this is a responder or this is a non-responder. And believe it or not, it took me 3 months to come with this. So we had to stay 3 months to try so many things and to really make sure if we're on the right track. Why? Because there's not a single correct criteria. People use things a bit differently. And thus, you need to really look at your data and to really make sure how you're going to decide this. So one way is the recessed criteria. This is a known imaging criteria. People use a lot in clinical, obviously. And it's simple. If you have your mouse and you treat it, if you have a complete response, it's a complete response. If the tumor shrunk by 30%, it's a partial response. If it grows more than 20%, it's a progressive disease. And some people just use recessed criteria for their PDXs. But the problem with this, and the question you would ask, does this take into account the intrinsic growth kinetics of each PDX model? In other words, do we know how was this tumor gonna grow anyways without the treatment? It could be that it's just a slow PDX and it was going to shrink anyways, because you can see how PDX developing it is difficult. Not all tumors take well, not all tumors grow well. So you're not sure if it was going to regress anyways with the treatment or without the treatment. So it's not the ideal way here. The better way is to do a tumor growth inhibition approach. And what you do is each PDX from each patient is grown in 2 mice. One would be the control and one would be your treatment mice. And from there, you can check the full change of the tumor. So you can check the tumor size at the end and divide it by the starting point and come up with the fold change for the control group. And then you can check for your treatment group endpoint versus the starting point. And you can use this formula. This is a modified formula that I've made based on the clinical cancer research paper. And from there, you get a tumor growth inhibition as a percentage. They've divided their groups into 4 groups: sensitive, if it's more than 100, because this formula just gives numbers that are more than 100, partial sensitivity or partial resistance, or resistant if it's less than 50%. So then you get more of a relative to your control. So then it's more relative to the control, it's more relative to the growth rate of this PDX in specific. Of course, you need to put on some limitations. So we've spoke that generating PDX is not easy. So you end up sometimes having just n equals 3 for each group. It's not easy to have a large number. So for example, if you see this LuCap35, this is a PDX. And if you see that if you have those 3 control not treated and 3 treated, and this is the tumor growth, you can see the majority are not growing that fast to start with, whether the control or not, but only one of your controls is growing. So this can skew the results, obviously, a lot. Some of these PDXs also end up dying early for whatever reasons as well. And you have sometimes the endpoint not very high. The timing of putting down the mouse is not very high. So you have an endpoint that's not very enhanced. So this can all skew some of your results. That's where we thought of 2 things here. First, to use the slope. So rather than using the end point versus the starting point, it's sometimes better to think of the tumor growth as a whole. So the entire trajectory of the tumor growth, the entire time of the tumor growth. And this is, I think, a more accurate way. It takes in account all the points. It does not take in account one point. It covers for a lot of the problems that we can see when you just measure the ending point. So this can be done easily. Prism or whatever calculates for you the slope for each mouse. And then you can use this slope as the entire tumor growth. And you can calculate slopes for your control or for your treatments. And another thing we've done is to make sure that it's really significant between groups. For example, as I told you here, LuCap35, only one sample seems to be doing the differences, but there's no actual difference between the control and treatment. So we wouldn't consider this a responder at all, while something like this, where all groups are showing differences, would be considered a clear responder. So we went for a strict criteria, and we said, if I would call responder, I had to have 2 criteria. For example, that the tumor growth inhibition is more than 80%, but also it's statistically significant between the groups. So this is a more stringent criteria. And as I said, there is no one way of doing it. So you need to really look at your data and see how it looks, because the most important thing is that you really want a responder group and the non-responder group. You don't want something that you're not sure of, because you're going to take this RNA sequence and then you want to find the signature. And just to show you, I will talk about this drug later, but just to show you the idea, so you can see here we were able to divide our PDXs into responders. So these are nice, clear responders that you can see the treatment suppressing completely the tumor growth. The blue here are the non-responders, so you can see there's not much difference between the control and the treatment. And somehow there are a group in the middle that are intermediates, and we did not include those when we come to do our RNA-seq analysis. So we focused more on the red and blue, or the very clear responders versus non-responders. So key message here, response criteria really defines your conclusion. So I think you really should choose them wisely. You shouldn't rush into anything. You should try a few things, and you should check really your data. You should check based on your numbers, and have like a clear methodological approach in this to justify for any paper as well, or for any reviewing. So now that we had our LUCAP series, as I said, we can test a lot of drugs. So I'll talk a little bit on the drugs that were tested here. So radiation is generally, prostate cancer responds well to radiation, but obviously in metastasis, you're not going to use radiation. You're not going to radiate everywhere in the body. But we use it more as a positive control, because we know that they respond a lot to radiation. But what we were interested in are two drugs, talazoparib and prexasertib. So talazoparib is a PARP inhibitor. And PARP is an enzyme that helps in single-strand break repairs. So it's a repair enzyme. Once you inhibit it, you're not going to be able to repair any single-strand breaks. And these develop into double-strand breaks. And obviously this cell cannot tolerate it much, and the cells die. So it's a used therapy in cancer. And in 2023, it was approved for metastatic castration-resistant prostate cancer. So it's already an FDA-approved drug. And people have shown that when used with androgen receptor inhibitors, it shows synergistic effects. And it has been approved for this, but only in patients who have homologous recombination repair mutations. So those who have BRCA1 or BRCA2 mutations. Because if I have a PARP inhibitor, but I have an intact homologous recombination repair, I will be able to overcome the double strand breaks. I can function well as a cell. But if I have a mutation in this, I'm not going to be able to do this. And this is what they call now in cancer synthetic lethality. If I have one that's not functioning, the other one might carry me. But if I have both not functioning, this is when it becomes a problem for the cancer cell, and it dies. So it was only approved for those. Recently, they've shown that it's not very beneficial for people who are not BRCA1 or BRCA2 mutated. But the one we were interested in more is the prexasertib. So prexasertib is a CHEK1, CHEK2 inhibitor. And this is also another DNA repair mechanism. So whenever this cell is under replication stress, CHEK1s are activated, and they basically tell the cell to calm down, wait, do not go into cell cycle, do not do anything, let us repair this damage, or if the damage is extensive, the cell needs to go into apoptosis. So by inhibiting this, obviously the CHEK1 is not going to be functioning, and then you end up with a lot of DNA damage. So it's also an interesting drug. Now, it took a little bit of a trajectory check, Prexasertib. At the beginning, people were quite excited about, but at some point, it has lost. It was COVID time, and the company that was developing it has lost a little bit of interest in, but it came back again. It has shown good results in ovarian cancer, in serious ovarian cancer. And it's been studied in some clinical trials as a single therapy or in combination. Interestingly, nobody has checked it yet in clinical trials or preclinical models strongly in prostate cancer. So we were really interested to test this drug. To show you some results, and this is unpublished data, so but we have published a little bit, of course, in still unpublished data, but we have published a bit in a lot of conferences, so it's okay to share at this stage these results. So we saw that with radiation you get 52% response in our PDXs. With the TALA, Zuparb, which is an FDA approved, we got 22%. But interestingly, with this drug that nobody has yet looked too much in depth in prostate cancer, we had 26% response. So that was exciting to see that it's doing well with some of our PDXs and even better than the FDA approval. Now, what was really also exciting to see, like all these rows are PDXs, so each column is a PDX, and rows are the treatment here. So if you look, for example, at PDX 145.2, you see, interestingly, it was a non-responder with the TALA, it was a responder with Prexa, and it was an intermediate responder with radiation. And you can also see that it was a neuroendocrine, so these are the type of the histology. So we said neuroendocrines tend to be the more difficult and more aggressive. And you can see it here nicely. This is the 145.2. So you can see it's responding well to Prexa. It's a bit of an intermediate responder based on our criteria. And it's a non-responder with the TALA. So this really shows that how we can use these PDXs and really compare various drugs and divide them into those responders or not. And it was really exciting to see that Prexa is doing well. It can give hope to a lot of patients who might be resistant to TALA or not responding as well for other therapies. Now, once we had this and we had responders and non-responders, we want to check how we can use the transcriptomics of these patients. So transcriptomics mainly means gene expression. So this is not genetics, but these are the RNA or gene expression signature. So I'm new, I'm still young in the bioinformatics world, but I'll take you through some of the things we've done. I'm sure some people might have much more knowledge in bioinformatics than I do. One of the known things for people who do some of the analysis is using the DSEC2 model. So this is used as an R package. And it basically gives me, I provide it with, these are my responder samples, these are my non-responder samples, these are all the RNA counts for all the genes that we've sequenced. And it can give me a list of differential expressed genes. So it tells me really what genes are different between those group and those group. It gives us something like this at the end. It gives us all the genes that we're looking for, a normalized mean count for each gene expression. It gives me a full change. So it's like telling me, your responder is expressing this gene 30 times higher, for example, than your non-responder. And it gives, at the end, this is the p-value that we look for, the p-adjusted value here. It's important to know that sometimes we avoid looking at very low counts. So people filter low counts, i.e. Genes that are expressed in very low amount should be filtered. There's different ways of filtering because if it's a very low amount, it might be different between responders and non-responders, but it's not very important or biologically important. So you might end up removing a lot of these free counts. Of course, each one of this needs a whole workshop to talk about. But once you have this list of your genes, differentially expressed genes, you can obviously do a lot of things. You can see which ones are upregulated, downregulated, what are the top genes that are different from each, and then we can do enrichment analysis. So it's taking these genes and saying which pathways are they most enriched in or found in, and you can get obviously different enrichment analysis done. And I recommend people who work in cancer to use mainly the hallmark gene set, which is gene sets that are related to cancer hallmarks rather than general function of this gene. So this makes it a bit better and more specific to see some of the enrichments. Now, another less commonly used transcriptomic analysis used, but not everyone maybe knows too much about, is the weighted gene co-expression network analysis. So it's a different type of analysis. What it does, it takes all your genes and it sees which genes are correlating with each other. So it divides them into modules where each module has correlated genes together. And this means that you're ending up with the pathways that might be similar or genes that might be functioning together, so they're correlating a little bit together. So, for example, you would have module 1 that has gene 1, gene 12, etc., because they correlate together, module 2. And this is a package also that does this all automatically on R. What's interesting, after you have these modules, so you can have 10 modules, 20 modules, 30 modules, after you do that, you can see, are they different between your responders or not? So R here are the responder samples, and R is the non-responder, the blue. And you can see, for example, in this module, there's not much difference, or it's only coming from one sample. But in this module, you can see that there is some difference. So this says that module 24 or whatever might be an important, the genes in module 24 might be important and might be different between responders and non-responders. So this is also another way we can analyze these transcriptomic data. So we started with all your genes, like 60,000, and we were able to do some differential expression or use the module system to get more limitation. Those 2,000 genes differentiate between our groups. But can we narrow it down further? And can we get more candidate genes and be more specific? At the end, you want a small signature. You want 10, 15 genes that might be different between our responders and non-responders. So one thing we can do is feature selection. And there are different ways of doing feature selection, but this was a package that we've used also in R. So feature selection uses statistical and machine learning methods. So it's a bit of an AI machine learning thing. What it does, it gives you the best genes. So it takes your 2,000 genes list, and through calculations and correlations with the response, it tells you, you know what, from those, and it removes the overlapping things or the extra redundant, and it gives you, you know, those are your most important genes that differentiate responders from non-responders. And we ended up really with these 42 selected genes. Unfortunately, I can't talk a lot about the genes because this is still unpublished data. There's some confidentiality in it. But you can see each column here, for people not familiar with heat maps, each column here is a sample. You have all your blue samples, not responders, or the red are the responders. Each row is your gene. And you can see this is the level of RNA expression. So nicely, you see a section here, section 1, that really differentiate your responders from non-responders, i.e. these genes here are more expressed in your non-responders, but are less expressed in your responders. So it seems to be like a good genes that can act as biomarkers, differentiating which people would respond to prexasertib and which don't. So you're limiting down and you're going from the list of 2,000 genes to more specific genes that seem to be doing the job. To further convince you with this, some people are familiar with PCA plots, but if not, it's basically how you cluster or categorize groups based on something. So if I use my entire RNA gene expression set, you can see that we're not able to cluster the groups. Everything is messed around. If I use my differential gene expression, so the list of the 2,000 genes or so, you see some clustering happening. So they really can separate the non-responders from the responders. But if I use the selected feature genes, so only these 42 selected genes, you can see that you start getting a better separation of your responder samples from your non-responder samples. So it really shows that these genes are really different between those two groups. What you can do even more to have an overlap between the two methods. So as I said, we use differential expression, DSEC, but we also use the module system. And sometimes you can look how many genes overlap between the two methods. And then you get up, even with the selected features, you can see what genes were overlapping between them. Because those show that these are genes that through two different methods are really showing the differentiation here. And again, by this heat map, you can see that you can get specific sections that really differentiate between your responders and non-responders. So it'd be interesting to look at those genes really and see what are they and how do they function. Are they related to any of the pathways related to PREXA? Now, I want to emphasize something. Not every gene could be important. For example, PSA, we all know PSA in prostate cancer. It does nothing in prostate cancer. It just happens to be a biomarker associated with prostate cancer, but not necessarily important in its biological function. So those genes might not be related to anything related to the PREXA pathway. They could, but they might not. So when you look for a biomarker, it does not necessarily mean that it will have a biological function or so. One thing also we can do is predictive models for tumor growth inhibition. So basically, we've divided our mice from the start into training samples and validation samples. We took the training samples and we did all these analyses, the RNA-seq analysis, the differential expression gene analysis, and we even divide them to training and testing samples. And then we use these differential expression genes to create a model. I've used the carrot package in R. So this is a model that really uses a lot of machine learning models, elastic net regression, lasso regression, different types of models we can test. And it just basically uses this RNA sequence to, and you give it which are your responders and which are your non-responders, and can predict if this patient will respond to treatment or not. So once you have these models, you can then test them on a validation sample. So these are untouched. It's like a different cohort that we haven't touched at all. And you can see if it will predict in these samples the treatment and non-treatment group. So to give you an example, these were the 29 that were the training, not the validation sample. These were the 29 training PDXs. This was the actual tumor growth inhibition. So 89%, this is a responder. 39 means that it was not a responder, and so forth. So these are the actual numbers after treatment. But once you train your system, these can predict for you the numbers. So these are the different models, and we've seen how it's predicting based on the RNA how much of the tumor growth will happen. So with the training sample, of course, it's going to be very close. It's very nicely predicting the numbers exactly as we expect. But the more important is the validation sample, the samples that it wasn't trained on. We can't say that it was the best outcome yet, because of course you're dealing with very small numbers. So the idea with training samples, you really need to have a larger number set, specifically so that some of these PDXs are from the same patient. So you would have this PDX 23.1, 23.1. It's the same patient, but from a different part of the metastasis. So the green boxes showed good prediction. So we can see that, for example, the elastic model was showing some good prediction with some of the mice, but not very good with some of the others. So it's still work under progress, but you can use these type of modeling to predict the outcome of your patients. So key message 4 here, you can use all these transcriptomic analysis to come up with the key genes and the gene signatures that can predict therapeutic responses. So these are a type of transcriptomic analysis that you can do. To finish up with the last 2 slides here, the question is this enough? And it's always not enough in science. There's always more to do. There's always things you can add, and there is no way out of a wet lab. So you really need to do your wet lab and do some screening and functional analysis. Bioinformatics analysis just tells you if there's an association, if this marker is associated with responders versus non-responders, but it's not really telling me more information or biological value of any of these genes or digging into the pathway. So you really need to do more things, specifically if you want to publish in high impact factors, and specifically if you want to know more about the genes and the characteristics of the genes. So a question would always be, are the genes in this pathway that I'm interested in truly important for any of the biological function and therapy response? And there are things you can do, but I'll talk quickly, like what we're doing currently in the lab, in Dr. David Labe, is the CRISPR-Cas9 library that he's studying currently. And this work is currently being conducted by Jason G to complement the bioinformatics work that I've done. So what they are interested in with PREXA is DNA damage repair genes. So they want to check if any of the DNA repair genes are important for PREXA sensitivity. And so what they do is they constructed a library. This is a DNA damage repair library of 1,800 genes. So they got any gene that is related to DNA repair, and they constructed a single guide. Companies obviously construct a single guide RNA. So that's a small piece of RNA that can bind complementary to this gene. These are then, they have already cell lines in the lab, prostate cancer cell lines, that express Cas9. And Cas9 is basically a scissor that cuts out any gene you want to cut using these single guided RNA. So if it sees the single guided RNA, it can cut here and you can knock out this gene. So libraries are constructed, so then you infect your cells so that each cell will get only one single guide RNA. i.e., each cell will only have one knockout of this gene. So this cell does not have gene X, this cell does not have gene Y, and we have 1,800 of these genes in the DNA repair. These cells are then treated with PREXA, or obviously a control, and grown for a few rounds, and then you can see which cells were responsive to the drug and which cells died, were sensitive, and which were resistant. And then you can sequence and measure your single guide RNA to know which knockout genes were important for responding or not responding to PREXA. So you start getting a biological value of this gene, i.e., if those cells that are resistant to PREXA means that they had a knockout of a gene that is important biologically for responding to PREXA. So this gives you more idea. And of course, this takes a lot of other works and optimization that I won't go through here just for the sake of time, but you require to know the IC50 for each cell line for your PREXA and know if it's really functioning in terms of the PREXA pathway, the phosphorylation pathway, and so forth. So this is obviously a lot of work, but it gives you an idea on how we use these DNA single-guided libraries related to a specific pathway. there is a whole genome library, so it includes all the genome, but usually people can do custom made to the pathway they're interested in. So for time, I think we're good on time. I want to give a different conclusion. So rather than saying that Prexa is good for prostate, might be an alternative therapy, that we're able to slowly reach a response signature related to it, we might be able to reach that. I want to really give a different conclusion here. So you can see that from the cycle again that we spoke about, to start with, for getting prostate cancer patients, you need clinicians to give us all the clinical data. Each PDX, each person, we have the entire clinical data of these patients. You need an autopsy pathologist to get the samples. You need the technicians. And once that's there and you want to establish your PDX, we see how the successful rate of PDX is not easy. So you need a strong animal facility, you need your scientist, and you need different drugs. Of course, we can test any combination of drugs, any drugs we want. I know in our part of this region, we are interested a lot in natural products as well. So we want a lot of these experimental drugs to test. And then we need a lot of bioinformatician to do some of these transcriptomic analysis and even more sophisticated analysis that could be done. And then once this is done, you go back in vitro and you want to study some of your genes, you want to do some functional analysis, or you want to do CRISPR-Cas libraries or so forth. So this requires also a lot of dedication and technical work. So my conclusion is it's time, I think, that we start thinking in Saudi Arabia and in our academic busy life for project managers. Academics are not the best in talking to each other. We're not the best in time management. We don't easily, everyone works a little bit on their own, and we don't have enough time, and we don't have enough, setting deadlines and tasks and baby tasks and managing things. We're not managers. So I don't see a lot of project managers in our labs. I've seen some, I have to admit, in Saudi Arabia, but we don't see a lot. And a project manager, of course, it's better if he has some science background, but also the most important is to really have leadership and managerial skills. And this helps to coordinate all these people. So a project manager would be able to coordinate these projects and really talk to different people and really to put the structure of the project together and to help things move on and move forward together to improve and to reach goals easier. I think it's something we should think about. I think it's something we should start seeing how we can recruit more project managers, how we can have them involved, how we can have in big laboratory settings project managers that can help in this coordination. I think it will help a lot. It will be a positive impact on enhancing some of this research. By this, I think this is the end of this talk. We made it in exactly 58 minutes, so I'm very pleased that we stuck to time. Thank you very much. I'm not sure how it works. I think there is a YouTube live, but I won't be able to really check it. Let me see if we can take... I think we can take some questions as far as I understand. So maybe if someone puts their questions in the chat, I can see if there are things I can catch because there's not a moderator. But if not, I would also like to share with you my email. So please feel free for anyone who has any discussions or questions. It would be very nice to have also collaborators that we can work with if they are interested. And if not, even feel free to come to the regenerative medicine unit, second floor in King Fahd, and we can really have discussions about any of this work. Okay. I'll keep it a few minutes. Thank you very much. I think everyone's on mute, so we cannot unmute people, I think. It might be difficult to unmute the large number, but feel free, as I said, to send any questions or any suggestions on the emails, and I'll be happy to really go into any discussions. Thank you very much, doctors. Thank you very much.